All right, Chapter 10, Skeletal Muscle Tissue. Four properties of skeletal muscle tissue. One, it's electrically excitable. This means that charge moves along the membrane of the muscle cell. This charge is called the muscle action potential. So you can see here the little dark arrow showing how the muscle action potential can move along the surface, the cell membrane of the muscle cell. Then there's contractility. This is the ability of muscles to generate force while pulling on attachments, aka the muscle gets shorter and pulls on a structure, causing movement. Extensibility is the ability of muscle tissue to stretch without being damaged, like a rubber band. And elasticity, which is it can return to its original shape and size, again, like a rubber band. Which of these is not a function of muscle tissue? Doobie-doobie-doo that. It would be secreting substances, B. So that means the other four, producing movements, maintaining posture, opening and closing body passageways, and producing heat, are all functions of muscle tissue. There are three main types of skeletal muscle tissue. There's three main types of muscle tissue. There's skeletal muscle tissue with very long columnar cells. These cells have these striated patterns all along their length, and they have multiple nucleuses per cell. These cells can be extremely long. They can be as long as the muscle itself. So sometimes they can be a foot or more in length. And they're found in skeletal muscles. Cardiac muscle tissue, found exclusively in the heart. These cells are much shorter than skeletal muscle cells. They branch, and they tend to only have one nucleus per cell. One nucleus, one nucleus, one nucleus. However, like skeletal muscle tissue, they do have that banding pattern, that dark light pattern running along their cells. They are involuntarily controlled. We don't have to think about making our heart beat. And between cells are these thick protein structures called intercalated discs. So between connecting the cells to each other, intercalated discs. Then we have smooth muscle tissue. Smooth muscle tissue is found in our hollow organs, such as intestines, stomach, trachea, and so forth. They are short little cells, kind of spindly in shape with the tapering ends. They have one nucleus per cell. Here's the nucleuses. And they are also involuntarily controlled. All right, muscles. Muscles have an origin, and that is the attachment onto a structure that is immovable. It's the side that does not move. And then they have an insertion. An insertion is to another structure, often a bone, skin, or cartilage, and that insertion site is where movement occurs. So in this case, with brachialis, when it has an insertion into the ulna, when it contracts, it'll move the ulna, aka move the forearm, bending at the elbow. A muscle organ is attached to a bone by a tendon. It is covered with a sheet of connective tissue called the epimysium. And this holds all these bundles of structures called fascicles. The fascicles are bundles of cells, muscle cells, and the fascicle is wrapped around by a sheet of connective tissue called the perimysium. And the cells, called muscle fibers, are each wrapped around by their own sheet of connective tissue called the endomysium. So we're going epimysium around the entire muscle organ, perimysium around the fascicle, and endomysium around the muscle fiber. And then within the muscle fiber, within the cell, we see these long protein rods called myofibrils. So we have bundles of myofibrils in my muscle fiber, bundles of muscle fibers making a fascicle, bundles of fascicles making the entire muscle organ. When you look at a muscle fiber, we notice there are various structures with names. The cell membrane, its name is changed to sarcolemma, so the plasma membrane is now the sarcolemma. The cytoplasm, its name is now changed to sarcoplasm. So sarcoplasm, the fluid inside the cell, sarcolemma, the plasma membrane. And sarcoplasm is a little different from normal cytoplasm because it has a lot of glycogen, which is a way to store glucose, and myoglobin, a protein that binds to oxygen. So muscle fibers have lots of glucose and oxygen stored within their sarcoplasm. And then, of course, as I already mentioned, these long protein rods that go the length of the muscle fiber, they're called the myofibrils, and they contract, they get shorter in order to make the muscle get shorter, and they're composed of protein filaments. Here's another view of a muscle fiber. We see some additional special structures for skeletal muscle cells. There are these tubes shown in blue, going through the width of the muscle fiber. Those are called transverse tubules. So they're just an invagination of the plasma membrane, the sarcolemma, that goes through the width of the muscle fiber. Also found within the muscle fiber are these membrane structures called sarcoplasmic reticulum. Normally at rest, sarcoplasmic reticulum is storing calcium ions. However, when it's time for contraction to occur, the calcium ions are released to signal that contraction. So sarcoplasmic reticulum are very important. When we look at the myofibrils, we see that they are made up of bundles of protein filaments called thick filaments and thin filaments, where the thick filaments are thicker than the thin filaments. And these filaments are formed a structure called a sarcomere. So one myofibril is made up of millions of sarcomeres, which are made up of thin and thick filaments. And it's the sarcomeres that get shorter during a contraction, making the entire myofibril get shorter. Here is another view of a sarcomere. Sarcomeres at their ends connect to each other by structures called Z-discs, so Z-discs at the end. In the middle of a sarcomere, they have a protein structure called an M-line. The Z-discs are what the thin filaments are attached to. The M-line is what the thick filaments are attached to. There are also regions within the sarcomere that have names. The A-band is the region where you have the thick filament. So the entire length of the thick filament is the A-band. The H-zone is the area where you have only a thick filament, no thin filament. So H-zone, only thick filament. And then the I-band is the regions where you have only thin filament, no thick filament. So I-band, only thin filament. Here's just another view of a sarcomere. Again, H-zone in the middle, A-band, the full length of the thick filaments, and the I-band where there's only thin filaments. And it turns out the sizes of H-zones and I-bands change as a muscle contraction occurs. So here is the sarcomere. Here is an electron scanning microscope view of a sarcomere. We can see the H-zones, the A-bands, the Z-discs, all the structures are there. The muscle is at rest. So we see a large I-band and large H-zones. And what's going to happen is the thin filaments are going to slide along the thick filaments, moving the thin filaments toward the M-line, the middle line. And that is how the shortening of the sarcomere occurs. So again, this is a relaxed muscle. When it contracts partially, the thin filaments are being pulled toward the M-line. As they're getting pulled toward the M-line, this causes more overlap between thick and thin filaments, also causes the sarcomere to become shorter. The H-zone gets smaller, the I-bands get smaller. Again, filaments are sliding, the thin filaments are sliding toward the M-line, shortening the sarcomere. When a muscle is fully contracted, we get complete overlap between the thin and thick filaments. There are now no I-bands and no H-zones. Of course, we still have the A-band because the smallest a sarcomere can get is the length of the thick filaments. That is how a muscle contraction occurs by talking about sarcomeres specifically. However, we want to know on a protein level how this works. So it turns out the thick filaments are actually made of millions of proteins called myosin. Myosin is a contractile protein able to bind to actin. And when it binds to actin, it's able to move the thin filaments toward the M-line, toward the middle of the sarcomere. The thin filaments, strangely enough, have three proteins on them. They have actin. Actin are represented by these gold blobs. They are the contractile proteins. They are what myosin binds to. So myosin on thick filament binds to actin on the thin filament. Then we have tropomyosin. Tropomyosin is these brown rods. Tropomyosin is a regulatory protein. It covers the myosin binding site. So at rest, it blocks myosin from binding actin. And then troponin, these little blue, I don't know if they are ghosts. Troponin is also a regulatory protein. It binds to calcium ions. And when it binds to calcium ions, it becomes able to change its shape. And when it changes its shape, it moves tropomyosin, thereby opening up the myosin binding sites. So tropomyosin binds calcium, then it changes shape. This moves tropomyosin, which reveals the binding sites on actin, allowing actin and myosin to get together. So this leads to what's called the contraction cycle. So what happens? Sarcoplasmic reticulum releases calcium ions. There's been a signal to cause a contraction. Sarcoplasmic reticulum releases the calcium ions. Calcium ions then come in and bind to troponin. Troponin changes shape and moves tropomyosin. We now have exposed binding sites on actin. So the first thing that happens is that we have the binding sites available on actin, and we have myosin ready to go with ATP attached to it. Myosin will take the energy from ATP, breaking it into ADP and phosphate, changing its shape. Myosin is now ready to bind to actin. Myosin will then bind to actin at the binding sites. This forms a physical connection between the thin filaments and the thick filaments. This is called a cross bridge. So by binding to the actin, there's now a physical connection between the thick filaments and the thin filaments. Myosin will then change its shape. When it changes its shape, it moves the thin filaments toward the M line. It shortens the sarcomere by moving the thin filaments toward the M line. This is called the power stroke and is the key cause of the contraction. And then finally, myosin will bind to ATP. By binding to ATP, myosin is then able to release actin. So ATP must bind to myosin in order to allow myosin to release actin. Then, assuming there are still plenty of calcium ions in the cell that are free, and there's plenty of ATP, this cycle can continue to go again and again and again, shortening the sarcomere over and over again until finally it gets as short as possible. Now, again, the myofibrils, those long protein rods that go the length of the muscle cell, are made up of millions of sarcomere. So when all those sarcomeres get shorter from the contraction cycle, this causes the myofibrils to get shorter, which then cause the entire muscle cell, the entire muscle fiber to get shorter, which then causes the muscle itself to contract. All right, so the signal comes from a neuron. A neuron sends a signal to the muscle fiber to say contraction must occur. So somatic motor neurons are required, and they form a connection with a muscle fiber at a spot called the neuromuscular junction. Neuro for neuron, muscular for the muscle cell. So that is where they connect. At the connection, the axon of the neuron widens out and is called a terminal bouton. The terminal boutons are filled with synaptic vesicles that have the chemical acetylcholine in them. Acetylcholine is a neurotransmitter. And the terminal bouton is then next to a certain region of the muscle fiber called the motor end plate. And then there's a slight space between the two structures called the synaptic cleft. What happens is the neural impulse arrives, the synaptic vesicles carry the acetylcholine to the synaptic cleft and releases acetylcholine into the synaptic cleft. Acetylcholine then diffuses across the synaptic cleft and binds to acetylcholine receptors on the motor end plate. When that happens, those receptors being bound to acetylcholine cause sodium ion channels to open. Sodium ion then floods into the cell. So this is positive charge flooding into the cell, changing the charge at this region at the motor end plate. If enough sodium ion channels are opened, then a muscle action potential will be generated. This will cause other sodium ion channels to open and close along the length of the sarcolemma, that's the length of the muscle's plasma membrane, and this charge will then move along the length of the muscle. So that is the muscle action potential, the opening and closing of ion channels that occur in one section, and then the next section, and then the next section, all the way down the length of the muscle fiber. So here it is. We get our muscle action potential at the motor end plate. This then causes the channels next to it to open. They then change the charge there, which causes the channels next to them to open, which then causes a change in charge there. So we get a movement along the length of the muscle fiber, sort of like the wave in a stadium where the cheerleaders get some people to stand up, and then they sit down, the people next to them stand up, and then they sit down, people next to them stand up, all the way along the stadium. And then the muscle action potential goes along the length of the muscle fiber and will go down into the transverse tubules. This change in charge in the transverse tubules signal the sarcoplasmic reticulum to release its calcium ions. So the muscle action potential in the transverse tubule causes the sarcoplasmic reticulum to release its calcium ions. And that leads to the contraction cycle and the end of this part of the lecture.